Hello, this is Maria El-Hokayem. Today I will be presenting the Benedicts test solution. Benedicts test is a chemical test that can be used to check for the presence of reducing sugars in a given analyte. Therefore, simple carbohydrates containing a free ketone or aldehyde functional group can be identified with this test using Benedicts reagent as an indicator. Benedicts solution contains copper 2 sulfate, sodium carbonate, and sodium citrate, and the copper 2 ions are blue. So with reducing sugars, they have free aldehyde or ketone groups that can donate electrons, reducing the Cu2 plus ions to Cu plus ions. When Cu2 plus is reduced to Cu plus, copper oxide forms, creating a colored precipitate. The color of the precipitate may be red, yellow, or yellowish-green depending on the nature and amount of the reducing agent present. Benedicts reagent is reduced by alpha-hydroxyaldehydes, alpha-hydroxyketones, and alpha-ketoaldehydes. It doesn't oxidize simple aromatic aldehydes. Moreover, molecules containing only the alcohol functional group or only the keto group are not oxidized by Benedicts solution. Since these groups lack the necessary structure to reduce copper ions in Benedicts solution, hence they don't produce a reaction. For this test, we will be needing a Bunsen burner, graduated cylinder, Benedicts solution, test tubes, test tube holder, and 2 beakers filled with butanol and acetone, where we will be using butanol as a positive control and acetone as a negative control. To start this experiment, we need to label our test tubes B for butanol, A for acetone. Moving on to the first step of this experiment, we start by adding 5 milliliters of butanol and acetone respectively to graduated cylinders, then add it to the respective test tube. Then, using Benedicts solution, which is a complex mixture of sodium citrate, sodium carbonate, and copper 2 sulfate, we will be adding 8 drops of this solution to each of the test tubes, A and B, and consequently noticing a change of color to blue in both test tubes due to the Benedicts solution. For the third and final step in this experiment, we will be using the Bunsen burner to heat the solution and indicate if any change of color happened or precipitate formed. Starting with butanol, we noticed a change in color to red, indicating a positive result. Moving on to acetone, while heating, we didn't notice a change in color, which indicated a negative result. The final results indicate a positive test for butanol, which was the positive control, as its color changed from blue to red, indicating the presence of reducing sugars, and a negative test for acetone, as the solution remains blue, hence there are no reducing sugars present. For more information, the book gave an example about testing sucrose with Benedicts solution. If we prepare a sucrose solution and add HCl and boil it, then neutralize the solution with dilute sodium hydroxide and add the Benedicts solution, we will notice that sucrose, which is a non-reducing sugar, won't initially react with the Benedicts solution. However, boiling with HCl hydrolyzes sucrose into glucose and fructose, which are both reducing sugars that will give a positive Benedicts test.